Hello everyone, I'm Dr. Fox, and let's continue our discussion of the cardiovascular system with a discussion of vasculature, or the vessels which distribute and return blood from and back to the heart. In this video, we'll describe the functions, physical characteristics, and features of arteries, arterioles, capillaries, and veins. We'll also define the various portal systems and give examples of both venous and arterial portal systems. Finally, we'll contrast systemic versus pulmonary circulation. So let's begin our study with that of the arteries. Arteries are defined as vessels that carry blood away from the heart. We typically assign them some sort of higher oxygenation status, and this is true in the systemic loop, but this is not true in the pulmonary loop, as we'll see. As they carry blood away from the heart, arteries typically enjoy a higher blood pressure. When distinguishing between arteries and veins in the laboratory, oftentimes arteries tend to be more tortuous. That is, they have a spiral or a curl to them. There are three basic layers of any vessel. We call them tunicas, or tunics as coats. There's the tunica interna, which is the innermost layer, the tunica media, and the tunica externa. So let's take a look at these in a little greater detail. So we'll start with the inside of the artery and we'll work our way outward. So the tunica interna consists of a very thin layer of endothelium. That endothelium is supported by a basement membrane, and that basement membrane is attached to a very thin layer of elastin protein that we call the elastic lamina. So this imparts a little bit of elasticity to the wall of the artery. As we make our way outwards, we come to the tunica media. The tunica media has two components. This first is the most robust and the most variable in thickness throughout all arteries, and that is the smooth muscle. The muscular layer of the tunica media is autonomically controlled. So the sympathetic and to a lesser degree the parasympathetic nervous system will have an effect on whether that muscle is constricted, therefore causing vasoconstriction, or relaxed, therefore causing vasodilation. The outermost portion of this layer is another more robust elastic lamina. And these elastic laminae, while they impart a little bit of elasticity, if you think about this in the context of blood pressure, as the heart sends blood forward, that stretches the walls of the artery, and these laminae help to bring the walls back closer together. And this will also impart force upon the blood to help drive it forward. The tunica externa is a mix of both elastin and collagen, and sometimes can include vasa vasorum, which are vessels that serve vessels. So these are smaller vessels that serve the walls of the artery. There are different types of arteries, but the general categories include elastic versus muscular arteries. Elastic arteries are considered conducting arteries. There are two fine examples of these: the aorta in all of its locations and the pulmonary trunk. These tend to be very large vessels, yet their walls are only about 10% of the total vessel diameter. They have very well-defined elastic laminae, but that muscular layer is not as great. And they serve as pressure reservoirs. So these are the arteries into which the heart is delivering blood directly. And so as it does so during ventricular systole, it stretches out the walls of these arteries and then they come back together. Hence, they're called elastic arteries. Elastic arteries are going to feed into muscular arteries, sometimes called distributing arteries. Muscular arteries, as their name suggests, have thicker tunica media, owing to thicker muscular layers within. and their walls tend to be about a quarter the size of their vessel diameter. These are the ones that are a bit more tortuous that we can see throughout the body. And they have a little more vascular tone to them than elastic arteries, and that is imparted by the autonomic nervous system. Muscular arteries are going to feed into smaller arteries, yet called arterioles, are resistance vessels. So the autonomic nervous system can control how much blood is flowing through them thanks to their very thick walls of smooth muscle. And when we think about autonomic control, there's the sympathetic division and the parasympathetic division. The sympathetic division predominantly will lead to vasoconstriction. Now, there are different types of receptors. There are alpha and beta adrenergic receptors, and these are influenced by norepinephrine and epinephrine. And typically, during a sympathetic response, there's vasoconstriction. And that's largely modulated by the alpha receptors. Alpha clamps down. But there are also beta receptors. Beta receptors are more stimulated by epinephrine than norepinephrine. And in tissues such as skeletal muscle, sympathetic stimulation can actually lead to vasodilation. And so there's this duality here. So the beta receptors broaden the smooth muscle. So sympathetic responses don't just do one thing, but they mostly do vasoconstriction, except largely for skeletal muscle. And in some cases, physiologically, cardiac muscle as well. And not cardiac muscle, the smooth muscle of the coronary arteries. These arterioles are significant in that they're going to lead into capillary beds. So you have arterioles feeding into things called metarterioles, and these pass through to venules. Now, what's interesting is that the metarterioles will have tiny little smooth muscle sphincters around the entrances to capillaries. We call them precapillary sphincters. And if they are relaxed, then the capillary beds are engorged with blood and there is a lot of exchange between the interstitial tissue. And if they are constricted, there is little to no exchange and the blood continues on past the capillary bed. So we have the ability of controlling the exchange of nutrients thanks to microcirculation, which is the distribution of blood through arterioles, metarterioles, and capillaries. Capillaries are thus exchange vessels, and they have a very simple structure. So think of an artery and strip away the tunica media and tunica externa and the elastic layer of the tunica interna. So you're left with just the tunica interna, the epithelial layer or the endothelial layer, and its basement membrane. Diameter-wise, oftentimes the lumen are only about 5 to 10 micrometers. And that's about the size of an average erythrocyte. And so erythrocytes go through capillaries one at a time. And so this helps us to maximize exchange of gases that way. On the other side of microcirculation beyond the venules, we have veins. And veins, as vessels, return blood to the heart. They have lower blood pressure, they are more thin-walled than arteries, and they still have the same 3 layers, the tunica externa, media, and interna, but they look a little differently. So let's take a look here. Let's start with the tunica externa because it is, well, exactly the same. It consists of elastin and collagen. It isn't any particularly different. The tunica media, however, lacks the elastic lamina, and while it does have some smooth muscle, it is a very narrow band. The tunica externa is also quite different in that it lacks the elastic lamina. It does have the endothelial layer and the supporting basement membrane, but sometimes that endothelial layer forms valvular structures, and these valves are a novelty which encourages the unidirectional flow of blood. So if blood starts to flow backwards, those valves can trap it and prevent it from retrograde flow. So venous flow is typically unidirectional. Not all veins have valves, but many do. And there are a couple of ways in which your body on the other side of the capillary beds with very low venous pressure can still encourage the movement of blood forward toward the heart. One of which is through a process called muscle milking. And with muscle milking, what happens is veins are going to be squeezed by adjacent skeletal muscles. And these adjacent skeletal muscles will squish the venous blood in one compartment, sending it through to the next compartment. And when the muscles relax, that blood cannot move backwards. So through the muscles squishing the veins, we have a means of advancing blood. Another way of moving venous blood forward is through respiratory milking. So we'll talk about this in the context of the respiratory system, but your diaphragm can change the volume of thoracic and abdominal cavities, and by changing the volume, it changes pressure. Changing pressure exerts pressure on organs and structures within, and as a result, that also puts pressure on veins, encouraging movement back towards the heart. Finally, and probably the one that I think is the neatest that I like, is the arrangement of veins as veni comitantes. Veni comitantes is the Latin way of saying accompanying veins. And sometimes you could have a single accompanying vein. That would be a vena comitans of an artery. Sometimes you can have two veni comitantes. These are always named for the arteries that they accompany. So for instance, the radial veins are veni comitanti of the radial artery. The brachial veins are the veni comitanti of the brachial artery, and so on and so forth. But what happens is these veins are adjacent to arteries. They're bound within the fascia which supports the arteries. And as arteries are pulsatile, that expansion of an artery puts pressure on the veni comitantes, thus encouraging the return of blood, much the same way muscle milking or respiratory milking would encourage the unidirectional flow of blood. It's really quite genius. What a wonderful thing to have evolved. Now, sometimes things go wrong. And within the context of the cardiovascular system, one might have a clot. So we call these thrombi, singular thrombus, and a thrombus is a clot inside a vessel, so a vascular clot. These typically remain stationary where they form, but they can dislodge and move. And if they dislodge and move and lodge elsewhere, we call this an embolism. So an embolism is a mobile clot that moves somewhere and can lodge elsewhere. So where this is most significant clinically are things called deep vein thromboses, or a singular deep vein thrombosis, a DVT. This may happen in any of the deep veins of the limbs or within the torso, or in the cranial cavity. But one of the most frequent places that this occurs, one of the more susceptible places, is in the lower limb. So the leg, the knee area, the thigh. And if a deep vein thrombosis forms there and it then embolizes, it can move up, travel along through the inferior vena cava, traverse the heart, and go out into the pulmonary arteries and obstruct the flow of blood. That deep vein thrombosis is called a pulmonary embolism at that point, if it lodges in a pulmonary artery. And that can be life-threatening because you're not able to move blood to that lung for gas exchange. So this whole phenomenon is venous thromboembolism, or VTE, but you probably have or will hear about DVTs and PEs. There are things that make people more likely to DVTs, so long periods of stasis, such as being on an airplane, any sort of vascular injury, any sort of hypercoagulopathy can encourage deep vein thromboses. So if you're traveling for long periods of time on an airplane, think of compression socks, think of getting up and moving around. If you have a job that requires you sitting for long periods of time, get up and move those leg muscles. Why don't you? Now let's look at portal systems, which are very interesting. So we'll discuss the caval system. The caval system consists of the superior and inferior vena cavae. We've talked a little bit about those already, and all of their tributaries. But we also have various portal systems, and portal systems are vascular systems where you have a capillary bed or more being drained by a vessel into other capillary beds, and that's pretty unusual. They may be arterial or they may be venous in nature, but let's examine some examples of these. One of the big ones that we'll discuss is the hepatic portal system. This is a venous portal system. Basically, all of the capillary beds of the gastrointestinal tract that absorb the nutrients from digestion are picked up in these capillary beds, and they're shunted via veins through the hepatic portal vein up to the liver for processing. There's also a hypophyseal portal system, which is another venous portal system connecting the hypothalamus and anterior pituitary, such that important hormones can be transported between the two. And then finally, an example of an arterial portal system is that of the renal portal system. And there are very many of these because we've got at least one per nephron, and there are millions of those in each kidney. So these are glomeruli. Glomeruli are specialized arterial beds that filter blood into filtrate, that then blood is conducted through efferent arterioles out to peritubular capillaries. And so we'll see why these are so important when we discuss the urinary system. So then we've got circulatory routes. The two major ones are systemic circulation and pulmonary circulation, and very many minor under systemic circulation. So systemic circulation is when we send blood from the heart out anywhere in the body and then back to the heart. So arteries carry blood away, veins carry blood back toward. Sometimes we give regional portions of the systemic route or loop names, such as coronary circulation for the heart, cerebral circulation for the cerebrum of the brain, the caval system, so all of the tributaries for the superior and inferior vena cavae, individual portal systems, so on and so forth. Pulmonary circulation is when we send blood from the heart to the lungs and back to the heart again. Pulmonary circulation is all about gas exchange with the lungs, whereas systemic circulation is many other things, but it's predominantly gas exchange with metabolically active tissues and other exchanges as well. So let's assess the knowledge that we've just discussed. Which layer of a blood vessel is the most affected by autonomic stimulus? Is it A, tunica externa? Oh, no, not so much. Tunica media, B, yeah, that sounds good. Or C, tunica interna? No, not so much. Tunica media, why? Because of the muscularis, or the layer of smooth muscle, is under autonomic control, and that is why B is the correct answer. Thank you so much for your time, and I look forward to talking to you in the next video. Take care, all.